Hello everyone. I hope you are all fine and doing great in your lives. I am your presenter Ismail and it's our second lecture. In this lecture we will talk about the organization of organisms. Firstly, I will discuss about the objectives of this lecture. In this lecture we will talk about the structure of a plant cell, structure of an animal cell, and we will also discuss the comparison in both structures. Comparison between prokaryotic and eukaryotic cells, functions of the structures, and we will discuss the levels of organization. So let's start our topic. What is cell? As you know that cell is the basic structural, functional and biological unit of all organisms. The word cell derived from a Latin word cella meaning small room. Cell is the smallest unit of life and cell is also known as the building blocks of life. In this diagram you can see that there are millions of cells. A human body is composed of millions of cells. Because the cell is the smallest unit of life and are also known as the building blocks of life. Now we will discuss the history of formulation of cell theory. So cells were first described by a British scientist Robert Hooke in 1665. In 1831, a British botanist Robert Brown discovered the nucleus in the cell. In 1838, Matthew Sheldon studied plant tissues and made the first statement of cell theory. In 1839, another scientist, Theodore Schwann, reported that all animal tissues are also composed of individual cells. Thus, Schleiden and Schwann proposed cell theory in its initial form. So, these are two scientists who proposed cell theory in its initial form. In 1855, Rudolf Virchow, who was a German physician, he proposed an important extension of cell theory. In 1862, Louis Pasteur provided experimental proof of this idea. Now we will discuss the three main principles of cell theory. First principle is that all organisms are composed of one or more cells. Second principle is cells are the smallest living things, the basic unit of organization of all organisms. And the third one is cells arise only by division in previously existing cells. So these three are the main principles of cell theory in its modern form. Now we will discuss the structure of the cell under light microscope. But firstly, we will talk about light microscope. What is light microscope? Light microscope is a microscope that works by passing visible light through a specimen. It uses two glass lenses. one lens produces an enlarged image of specimen and the second lens magnifies the image and projects it into viewer's eye or on a photographic film. So it has the two glass lenses. One lens produces an enlarged image and the second lens magnifies the image and projects it into a viewer's eye or on a photographic film. Now we will talk about the micrograph. What is micrograph? A photograph taken through a microscope is called a micrograph. A light microscope can magnify the objects only about 1500 times without causing blurriness. Its magnification is 1500x and its resolving power is 0.2 micrometer, and you know that 1 micrometer is equal to 1 over 1000 millimeters. Now we will discuss about the cell structures under light microscope. So when we study the structures of the cell under light microscope, we will find out the organelles. So the organelles which can be seen under light microscope are nucleus, cytoplasm, cell membrane, chloroplast, cell wall and mitochondria. So mitochondria is also visible under light microscope, but detailed study of the mitochondria is not possible under light microscope. Now we will discuss about the animal cell structures under light microscope. Using a light microscope, we can see cell walls, vacuoles, cytoplasm, chloroplast, nucleus and cell membrane. So these are the structures of the animal cell which are visible when we see the structure of animal cell under a light microscope. Now we will talk about the plant cell structures under light microscope. Under light microscope we can see cell walls, vacuoles, cytoplasm, chloroplasts, nucleus and cell membrane. So these are the structures of a plant cells which are visible under light microscope. Now we will compare the plant cell and animal cell. Animal cells do not contain chloroplast while plant cells contain chloroplasts. Animal cells are not surrounded by cell walls while the plant cells have the cell walls. The vacuoles in the plants are much larger than those of animals. So the vacuoles in the animals are smaller than the vacuoles in the plant. This diagram shows the difference between the animal and plant cell. As we can see that plants have the cell wall, large vacuole, chloroplast and flagella only in gametes present, while in animals no cell wall is present, small or no vacuole, no chloroplasts and flagella is present. Both of these have mitochondrion, Golgi apparatus, rough and smooth endoplasmic reticulum, nucleus, cytoplasm and ribosomes. So this is the basic comparison between plant and animal cells. Now we will discuss about the prokaryotic and eukaryotic cells difference. Nucleus is not present in prokaryotes while it's present in eukaryotes. Membrane-bounded organelles are absent in prokaryotes while present in eukaryotes. Size of a prokaryotic cell is 1 to 10 micrometer, while in eukaryote it is 10 to 15 micrometer. Prokaryotes were evolved 3.5 billion years ago, while eukaryotes were evolved 1.5 billion years ago. Cytoplasm is present in prokaryotes while it's also present in eukaryotes. Cell membrane is also present in both of these, while cell wall is present in prokaryotes but it is present in plants while absent in animals in eukaryotes. Ribosomes are present in prokaryotes as well as in eukaryotes. DNA in prokaryotes is in circular and free-floating form, while in the case of eukaryotes, there are chromosomes in the nucleus. Examples of prokaryotes include bacteria, while examples of the eukaryotes are plants, animals, fungi, and protists. Now we will talk about cell wall. Cell wall is a non-living and strong component of the cell that is located outside the plasma membrane. Animals and many animal-like protists do not have the cell wall. The outer layer of the plant cell wall is known as the primary wall, and the cellulose is the most important component. Fungi and many protists have cell wall. Although they do not contain cellulose, their cell walls are made up of a variety of chemicals. For example, chitin is present in the cell wall of fungi, while prokaryotes have a cell wall that is composed of a chemical known as peptidoglycan. If we talk about the functions of a cell wall, then cell wall provides the shape, strength, protection and support to the inner lining matter, which is known as protoplasm of the cell. So protoplasm is the inner living matter of the cell. Cell wall also prevents the drying up of the cells, and it also helps in controlling the cell expansion. Now we will discuss about cell membrane. All prokaryotic and eukaryotic cells have a thin and elastic cell membranes covering the cytoplasm. It is mainly composed of proteins and lipids with small quantities of carbohydrates. Functions of the cell membrane includes: cell membrane functions as a semi-permeable membrane. It allows a few molecules across it while functioning the majority of chemicals inside the cells. Cell membrane maintains the internal composition of the cell, and in addition to this role, cell membrane also sends the chemical messages and can identify the other cells. What is cytoplasm? Cytoplasm is the semi-viscous and semi-transparent substance between plasma membrane and nuclear envelope. It contains water in which many organic molecules like proteins, carbohydrates, lipids, and inorganic salts are completely or partially dissolved. Functions of the cytoplasm include: it provides space for the proper functioning of organelles. It also acts as a site for various biochemical or metabolic reactions. For example, glycolysis. Glycolysis is the breakdown of glucose during cellular respiration that occurs in the cytoplasm. What are ribosomes? Ribosomes are the smaller granular structures that are either floating freely in the cytoplasm or are bound to the endoplasmic reticulum. Each ribosome is made up of almost equal amounts of proteins and ribosomal RNA. They are not bound by membrane and also found in prokaryotes. Eukaryotic ribosomes are slightly larger than prokaryotic ribosomes. Functions of the ribosomes include: these are the sites for the protein synthesis. Protein synthesis is very important for a cell. When a ribosome is not working, it is disassembled into two smaller units. What is mitochondria? Mitochondria, in singular form known as mitochondrion, these are the double membrane bounded structures found only in eukaryotes. The outer membrane of a mitochondrion is smooth, but the inner membrane forms many enfoldings called cristae, singular crista, in the inner mitochondrial matrix. Mitochondria have their own DNA as well as ribosomes. If we talk about the functions of mitochondria, these are the major sites of aerobic respiration and are the more energy producing centers. Nucleus is a prominent structure that is found in eukaryotic cells. So nucleus is prominent in eukaryotic cells. In animal cells, it is present in the center, while in mature plant cells, due to formation of large central vacuoles, it is pushed to side. It is bounded by a double membrane which is known as nuclear envelope. Nuclear envelope contains small pores that enable it to act as a semi-permeable membrane. Inside nuclear envelope, a granular fluid which is known as nucleoplasm is present. What is nucleus? It is a dark spot and it is the site where ribosomal RNA are formed and assembled as ribosomes. Chromosomes are visible only during cell division, while during interphase they are in the form of fine thread-like structures known as chromatin. The prokaryotic cells do not contain prominent nucleus. Their chromosome is made up of DNA and is submerged into cytoplasm. What is endoplasmic reticulum? It is a network of interconnected channels that extend from cell membrane to nuclear envelope. It exists into two forms, rough endoplasmic reticulum and smooth endoplasmic reticulum. What is rough endoplasmic reticulum? Rough endoplasmic reticulum is called rough because of its rough appearance due to numerous ribosomes that are attached to it. And because of this large number of ribosomes, it serves as a function in protein synthesis. Now we will discuss about smooth endoplasmic reticulum. It lacks the ribosomes and it is involved in lipid metabolism and in the transport of materials from one part of the cell to the other. It also detoxifies the harmful chemicals that have entered the cell. What are vacuoles? Vacuoles are the fluid-filled single membrane-bound organelles. Cells have many small vacuoles in their cytoplasm. However, when a plant cell matures, its small vacuoles absorb water and fuse to form a single large vacuole in the center. Cells in this state become turgid. Many cells take in materials from outside in the form of food vacuole and then digest the material with the help of lysosomes. Some unicellular organisms use contractile vacuole for the elimination of waste from their bodies. Now we will discuss about chloroplasts. Chloroplasts are the organelles found in plant cells and eukaryotic algae that conduct the photosynthesis. Chloroplasts absorb sunlight and use it in conjunction with water and carbon dioxide gas to produce food for the plant. And it is the process of photosynthesis. Now we will talk about the levels of organization. First level is atomic and subatomic level. Then molecular level, macromolecule, cell level, tissue level, organ system level, organism, population, community, ecosystem and biosphere. Now we will discuss each of these levels in detail. First level is subatomic and atomic level. As you know that all type of matter are made up of elements and each element contains a single kind of atoms. These atoms are made up of many subatomic particles. The most stable subatomic particles are electron, proton and neutrons. What is molecular level? Bioelements combine through ionic or covalent bonding. The stable particle formed by such bonding is called as molecule or biomolecule. There are two types of the molecules. Number one is macromolecules. Macromolecules are those with low molecular weight, for example glucose and water. Second category is macromolecules with high molecular weights, for example starch, proteins and lipids. What is organelle and cell level? Biomolecules assemble in a particular way and form organelles. These organelles are subcellular structures and when they assemble together, they form unit of life like cells. As we have discussed earlier, the cell is the basic unit of life. For example, prokaryotic cells and eukaryotic cells. Each type of organelle is specialized to perform a specific function. For example, mitochondria is an organelle. Now we will discuss about tissue level. In multicellular organisms, similar cells which perform similar functions are organized into groups which are known as tissues. For example, epidermal tissue, ground tissue, muscle tissue and many more. What is organ and organ system level? In higher multicellular organisms, more than one type of tissues having related functions are organized together to form an organ. For example, stomach is an organ. Different organs which perform related functions are organized together in the form of an organ system. For example, digestive system is an organ system. In an organ system, each organ carries out its specific function. What is individual level? Different organ and organ systems are organized together to form an individual or organism. What is population level? A population is defined as a group of organisms of the same species located at the same place in the same time. For example, the population of any country in 2016, 2018 or anything. So it is the example of a population. For example, the population of America in 2016. What are species? Species is defined as a group of organisms who are capable of interbreeding and they produce fertile offsprings. For example, human beings are the example of species because they are capable of interbreeding and produce fertile offsprings. What is meant by community level? Community is an assemblage of different populations which interact with one another within the same environment. For example, a forest is considered as a community in which different populations interact with one another. What is biosphere? Biosphere is the part of the earth that is inhabited by organisms communities is known as biosphere. It constitutes all ecosystems. What is ecosystem? Ecosystem is an area where living organisms interact with the non-living components of the environment. And it is also called as the zone of life on the earth. So it is biosphere. Now we have discussed all the contents of our lecture. I hope it helps you to understand all the topics. Thank you.